Okay, so I'm ready to get the supplies that I need to carry out the ligation and transformation lab. So we're going in here to Life Technologies. This is a good company. It has the stuff that we want. So it takes a second to open this. Then I'm going to go in here and I'll search for what I want. I want the topo TA cloning kit. Okay. So here's our topo TA cloning kit, subcloning without competent cells. So that's actually the one that I want. But I'm going to show you this one first. If you have plenty of money, then you want this one because it has chemically competent cells, this top 10. You can basically do your cloning in about 5 minutes with this, whereas the other one, we have to get the E. coli and we have to make them competent. So it takes more like 35 minutes. So this brings in E. coli. You have to keep this E. coli in negative 80-degree freezer. We have used this in the class in the past, and it hasn't worked very well because of the delicate nature of the bacterial cells and the vector in here. Okay. So instead, what we're going to do is we're going to go in here and get this topo TA cloning with competent cells. You'll notice it's way less expensive. So $237 for 10 reactions. I guess only about $100 less expensive. So this is the one that we want, and we'll grab this one then for the class. Beyond that, then I want XGAL, an XGAL solution for our blue-white screening. Okay, and I have several options here. I actually want this option down here, so we'll look at this one. So here's our XGAL solution right here. It comes in 222s for this 1 gram of XGAL solution. I actually want it in solution. So I think we'll go here to this XGAL ready to use instead. 10 mLs, 131, that's what I want. So I'm going to get that also for the lab. Okay, so one of the tough things about microbiology is that everything has to be sterile. This is our sterilizer called an autoclave. You guys will be actually using this during the first in-person class on media making, where we have to autoclave the media to make sure that the media is all sterile. Okay, instructions are up here. It says to operate, fill with water, load the sterilizer, close the door, select the exhaust, and set the timer. So the first thing we have to do is load this with water. So I'll basically close that there, hook this water up to our hose up to our water right here, and I'm going to load the sterilizer with water. Okay. There's a little line down here. This line basically says fill just below this line. Okay. I'm going to fill up the sterilizer till it is just below that mark. All right. Okay, now I want to load the sterilizer. I have a lot of stuff that we need for this lab. So here is our transformation calcium chloride solution. Made up a rack of luria broth now that needs to be sterilized. We have to sterilize all of our pipette tips. So we have to sterilize our 1000, our 200, and our 100 microliter pipette tips. We also need microcentrifuge tubes. And you always cover things with aluminum foil if you can, because aluminum foil can work in the sterilizer. And aluminum foil, then when we take things out, little bacteria don't fall in it. So you want to cover things with aluminum foil if you can't cover them in another way. Okay, close the door. And then we have to set exhaust. Here is fast and slow. Fast is for instruments, slow is for liquids. We do have 1 item of liquids in there. Okay. Key thing for this is that this is basically works on pressure and temperature. The pressure builds up inside, the temperature builds up inside. We'll be going up to 250 degrees Fahrenheit or 121 degrees centigrade. This right here is a pressure indicator. It'll indicate the pressure when it's fully in and on. And basically then through pressure and temperature, then you're going to kill all the bacteria inside. Problem is, once pressure is built up, if we release that pressure, if you release it too fast, then solutions go and become gaseous and everything bubbles over. Okay, so generally when you use the autoclave, you don't want to fill things more than half full. And then you want to make sure you're on a slow exhaust if you have any liquids. If you're on only solids, you can use fast exhaust because then you don't have to worry about the bubbling over and the gaseous state of that liquids. And I basically turn this on now for 15 minutes, and then I will leave it for 1 hour. All right, so an hour has passed, and so now we want to check this out here. So if you look up here, we look at our pressure gauge. Our pressure is down. This actually is an indication of when you can't open the door. It's below where we can open the door, so that's good. And of course, because this is super hot, I need my oven mitts. And you have to be careful because sometimes really hot stuff drips right there. But basically then we just have to take everything out of here. Okay. And it's all nice and hot. All right. And that's it for the autoclave. Okay, now I've got to take all this stuff and use it in the experiment. Okay, so I got the Topo TA cloning kit right here. Key thing we want is this PCR 2.1 Topo vector. If we look inside this cloning kit, then we can see a lot of stuff. Here's all the stuff we would need for PCR. So there's our DNTPs, water, PCR buffer, control PCR template, forward, reverse primer. So if I was running a PCR, I would go in, take the DNA, run the PCR, and then I would put it into the vector. Since we already have our PCR product, all we need is this vector right here. So a little teeny bit of that vector then is what we have to use for our ligation. Okay, so I'm going to work through procedure number 1 on page 83 of your lab manual. Step number 1 is basically to get an LB auger plate with ampicillin. And so here's an LB auger plate. The plus on the bottom means that it has ampicillin. So we basically made regular LB auger here and then added ampicillin after it cooled to about 50 degrees, which is important because if you don't let it cool, then you actually denature the ampicillin. So it's not the easiest plate to make. Okay. And so our second step then is basically going to add 20 milligrams per ml of X-Gal. So here's the X-Gal that I purchased, ready to go. Okay, and I want to add 40 microliters of that X-Gal solution to the plate, and then I want to spread it over the plate and prepare it for use in the lab. So first step then, I have my pipetter. It's set to 40 microliters. We'll get that. We'll get our X-Gal solution out. Add our 40 microliters of our X-Gal here. Get rid of my pipette tip. Okay. Now I'll go ahead and light the Bunsen burner. All right. I'm upstairs now in the Gem Bio Labs instead of the Microbiology Labs to do this. So I didn't have a spreader on hand. I didn't want to run downstairs, but I just grabbed a paper clip out of my desk and I adjusted the paper clip. And so I'm going to heat up the paper clip here over the Bunsen burner so that it's nice and hot. I need to cool this off to make sure that it's not too hot. So I just grabbed a different plate of auger to cool off my spreader. And then I'm going to take this spreader then and basically really spread it around this plate so it's nice and evenly spread around this entire plate. Okay, I'm going to return that. I'll do this and then I'll add a label to the plate for the appropriate treatment that we'll use. And I'm going to throw the plate in the incubator until use. Okay, so now we're ready to go on to step number 3 in procedure 1. And that's basically adding our topo vector, 5 microliters, to 1 microliter of our PCR product. So here's our topo vector from the kit. Here's our PCR product that we just used in electrophoresis. All of these gave us good results on our gel. So I'm just going to grab the first one here, which is AE2, and we'll put it over here for AE2. I will grab out a microcentrifuge tube. This just came out of the autoclaves. These are all sterile. Make sure we close that lid on the microcentrifuge tube so that it remains more sterile in there. I want 5 microliters of my topo vector. I'll start with the largest volume first. Take a pipette tip. We'll grab our vector right here, and we'll go in and carefully get 5 microliters of that vector. Okay. And then I will put the 5 microliters of the vector down in the bottom. Make sure that drop stays in there, which it does. And then I'm set for that. And we'll get rid of the pipette tip. Now we have this one set to 1 microliter. So I'll go here, get another pipette tip. We'll go into our AE2. And we'll go in very carefully and get 1 microliter out. It's a teeny tiny drop in there. And then we'll put it into that tube, and see, I'll mix it up here so that we get it, and then make sure that drop stays in the bottom. And we are all set, ready to move on to the next. All right, step number 4 is adding calcium chloride to a second tube, number 2. So this is a sterile microcentrifuge tube that I just grabbed from the jar that we got out of the autoclave. I labeled this one number 1. This was our ligated vector in here. Okay, so I'm just adding the cold calcium chloride to tube number 2. Okay, so now I'm set there. Okay, so now steps 4 and 5 are next. So what I have here is a plate of E. coli. This I made 24 hours ago, and I incubated it at 37 degrees centigrade. Here we can see each of those little dots is a colony. We basically want to take 3 colonies, put the 3 colonies in our ice cold calcium chloride, which is then going to make them competent. And then to the ice cold calcium chloride and the E. coli, I want to add our ligated vector to that, and so that if these are competent, then they're going to take up the ligated vector. Okay, so to start with them, we'll light the Bunsen burner. I'll take my loop and I'll flame my loop so that it's nice and red hot here. So you can see that. Sorry, you can't see the top of that, but I keep it in a downward motion. You can see how hot that is. Okay, then I'm going to take my plate, and my loop's really hot, so I'm going to cool my loop off on a spot on the plate there to the side. Okay, once my loop is nice and cooled off, I'm going to take 3 colonies, so 1, 2, 3. Nice. So here are my 3 colonies. I'll take my tube number 2 with my ice cold calcium chloride. Put my colonies of bacteria in here so that the ice cold calcium chloride can start working on these cells and making them competent. Okay. And when I'm done with that, I want to flame my tube here. Now what I want to do after my tubes are all flamed, we'll turn that off. I have this set at 6 microliters, so I'm going to take 6 microliters of my ligated vector now. Okay, and I'll grab all 6 of those. That's pretty much everything in there. Very teeny tiny drop. And we'll put all 6 microliters in here, and I'll basically mix it all up, make sure I get that out of that pipetter there so that it's all outside. And then I'll place this on ice for 15 minutes. Okay, so 15 minutes is up. So I basically want to transfer my microcentrifuge tube to water bath. Right there, perfect, 42 degrees. Okay, here's my floaty. I'm going to take my microcentrifuge tube, put it in the floaty, and float it in there for 90 seconds. Okay, so my 90 seconds are almost up. And when my 90 seconds are up, then I'm basically going to take this out, and we'll take that microcentrifuge tube, put it back on ice then for 2 minutes. Okay, let's see, my 2 minutes are almost up. So at 2 minutes, then I'm basically going to take the luria broth, and I'm going to add 250 microliters of luria broth to my culture. Okay, and then I'm going to move it over here, flick it a little bit, mix it around, and leave it there at room temperature for 15 minutes. So 15 minutes have passed, and here is our sample. So we have in here then luria broth. Inside that luria broth we have E. coli. The E. coli has been transformed by the ligated plasmid, and the ligation was from our PCR product, from our AE2 PCR. Over here we have our plate. This is a plate we did earlier. I labeled it AE2, and this plate has Xgal on it, all ready to go. So the first step then is to take 50 microliters of our sample here, and I'll basically mix it up nicely so that we get a good sample. And we'll take those 50 microliters and put them on our plate. Now the next step then is to spread that around, so we're going to light the Bunsen burner. And I'm going to use the paper clip again because it worked well last time, so I'll basically heat it up on this Bunsen burner so it's nice and hot. You can see that, so nice and red hot there. I'll cool it off on this plate again like we did last time. Okay. I'll move this out of the way, take this guy, and we'll basically go in and stir that all around. I'll move it this way, so I like to go that way. And we'll stir that all around so that it's nice and smooth across the bottom there. Okay, and fully spread out everywhere. Alright, we'll reflame our paper clip, make sure we kill everything on there. And we're ready to go. So I'm just going to go throw this in the incubator for 24 hours, and then we will see what we get tomorrow. Okay, so 24 hours have passed, and we'll go in and check and see what happened to the plates. So let's see here. We have our no transformation on ampicillin plate. Looks like no growth. Ooh, a lot of growth. No transformation on no ampicillin plate. No ligation on an ampicillin plate. No growth. Okay. No, actually, it does have a little bit of growth in there. So very, very small ones. If I left these 48 hours, it would be bigger, but that one actually looks pretty good. Those are looking blue like they should. Okay, good growth on this one, our AE2, and good growth on our B2. So it looks good, looks like it's successful. Now I'm going to go in and we'll look at all of these a little bit more in detail. Okay, so I just want to look at several of the plates here. Okay, so here is one control that I ran. This is a no ampicillin plate, and I just ran the E. coli through the procedures that we did through the calcium chloride and things without actually transforming them. So these are untransformed bacteria, and they worked really well. So we can see everything there. And if we put it on white, we can see that it's all white plate. Okay. The next plate then that I ran is a no transformed plate with ampicillin. And we can see here no blue colonies at all, and we can see here no growth. So if they didn't get transformed, then they didn't grow, so we didn't have any ampicillin resistance. Then I ran an interesting one where I basically did not put the PCR product in. And so without the PCR product, then that PCR product couldn't ligate into the plasmid. So this is the plasmid straight without the PCR product. Okay, remember we put XGAL on. XGAL then, if that plasmid was intact, would turn blue. And if you look at these, all of those, we can't see them super clearly, but all of those are blue. So I don't see a single white colony on there. All blue colonies on there. If we can check that out. Okay. All right. So next then, let's look at our actual ligated plates. So this one was ligated with the PCR product from AE2. And we can see all of those are white. Looks like a couple blue colonies. So almost all white, which is what we would expect if the ligation worked. In here we can actually see a couple blue colonies. So see that blue one there and that blue one there. Okay. So almost all of them, the ligation was successful and the transformation was successful. Okay. Here then is our final plate. I did also the same thing for the B2. So here's a PCR product ligated in with the PCR product from B2 primers. And again, almost all white, very successful, growing on ampicillin resistance plate or ampicillin plate. And we can see only a couple there didn't ligate. So the ligation step worked really well. We were highly successful with that. So then the next step in the process, in this case, we would then take these colonies out, put them in a 90 well plate, and grow them up and then send them off to be sequenced.